welcome to today's video lecture. In today's video lecture, I'll be solving previous year's NEET questions and discussing the answers. And this is for the chapter Biomolecules, and we are in part 2 of the series. So if you haven't checked out part 1, please do so. Alright, so let's begin. Question number 1. Which one of the following is the most abundant protein in the animals? And this appeared in NEET 2020, pretty recent. A. Hemoglobin, B. Collagen, C. Lectin, and D. Insulin. Okay, so we're looking for the most abundant protein in the animals, and that is B. Collagen. Alright, so your answer is B. Let's go to the discussion. Alright, collagen is the most abundant protein in the animal world. It contains 4-hydroxyproline and 5-hydroxylysine. So these are the two amino acids that are present in collagen. Okay, it is a connective tissue, as we all know, and forms a ground matrix substance in cartilages and bones. All right, so I've already discussed the synthesis of collagen previously in structural organization in animals, so you can check that out. And yeah, to answer this question, yeah, it was pretty direct. Collagen is the most abundant protein in the animal world, and it is made up of amino acids, and yeah, it contains proline and lysine. Okay, so you remember these amino acids. They could potentially ask you this question, I'm not sure, but we should be aware. Right, this was pretty direct, right? So let's move on. Question number 2. Which of the following glucose transporters is insulin dependent? Okay, insulin dependent, and this appeared in NEET 2019. Again, pretty recent. A. GLUT4, B. GLUT1, C. GLUT2, and D. GLUT3. So which one of the following glucose transporter is insulin dependent? And it is A. GLUT4. Okay, so the answer is A. So GLUT4 is an insulin dependent glucose transporter. It is present in skeletal muscles and adipose tissues. Now, insulin increases the number of these transporters and promotes the activity of GLUT4 in skeletal and adipose tissue. So if you remember, in the endocrine system, I discussed the function of insulin and how insulin transports the glucose into the skeletal tissues and the adipose tissues. Okay, so there are receptors present. Let's say this is a skeletal tissue. There are some receptors present over here. Okay, so these are insulin dependent receptors. Okay, so like I said, over here insulin increases the number and promotes the activity of GLUT4. Okay, so these are GLUT4 receptors. Okay, so let's say if there's a lot of glucose in the blood. Okay, so in pancreas secretes insulin. Okay, and what's the job of insulin? To transport these glucose into these skeletal tissues. Okay, skeletal muscle tissues, and you need receptors. So okay, and so these GLUT4 receptors are dependent on the amount of insulin that is secreted from the pancreas. Okay, so yes, GLUT4 is an insulin dependent glucose transporter. So yes, when there's glucose, insulin transports these glucose molecules to the receptors, which then go into the skeletal tissue and provide energy. All right. Okay, let's move on then. I guess we've answered this question. GLUT4 is there in NCRT, but they haven't really specified, you know, whether it's an insulin dependent or not. But yeah, this is a good question. You should know that GLUT4 is the insulin dependent glucose transporter. All right, let's move on then. Question number 3. Which one is the most abundant protein in the animal world? I think we already discussed this before. This appeared in 2012. Again, a repeat of the same question. A. Trypsin, B. Hemoglobin, C. Collagen, and D. Insulin. Okay, so the most abundant protein in the animal world is C. Collagen. Alright, answer is C. Yep, I will not discuss this again because this is just a repetition of the first question. And we'll move on then. Question number 4. Which of the following have carbohydrate as prosthetic group? Okay, this appeared in 2000. A. Glycoprotein, B. Chromoprotein, C. Lipoprotein, and D. Nucleoprotein. Which of the following have carbohydrate as prosthetic group? Clearly, carbohydrate, you can see from the name itself, it's glycoprotein. Okay, glyco is the prosthetic group. So the answer here is A. So the discussion. So when several proteins are covalently bound with carbohydrates, it forms glycoprotein. So here are some of the examples of the different glycoproteins that are present in nature. There's collagen, there's immunoglobulins, fibrinogen, all these are examples of glycoproteins. Okay, so you have like a protein part and you have a carbohydrate part. Okay, so you have a protein part, you have a carbohydrate part, together they form glycoproteins. Okay, pretty simple from the word itself, glyco, so you know that they're talking about carbohydrates. But it's important to remember that these are also examples of glycoproteins. They can ask you, what is immunoglobulins, what is fibrinogen, what is collagen. So these are types of glycoproteins. Okay, let's move on then. Question number 5. Which one of the following statements is wrong? Okay, let's emphasize on wrong. NEET 1 2016 is when this question appeared. A. Uracil is a pyrimidine. Okay, that's correct. B. Glycine is a sulfur containing amino acid. That's incorrect. C. Sucrose is a disaccharide. Correct. Cellulose is a polysaccharide. Yes, that's also right. So the answer is B. So glycine is a simple amino acid containing no variable group. So we already discussed which is the simplest amino acid. There's a question asked previously in part 1 also. Glycine is the simplest amino acid that is there. It contains no variable groups. It contains just one hydrogen atom here. Okay, so it's glycine. Okay, this was also pretty simple. You just had to know, yes, you had to be aware of the different types of carbohydrates that are there, and the bases and the amino acids. You had to be aware of that. But they were pretty simple statements. So I guess we can move on then. All right, so question number 6. The chitinous, I think it's chitinous exoskeleton of arthropods is formed by the polymerization of, and this appeared in 2015. So we already discussed cockroaches, the structure of cockroaches and structural organization of animals. They have chitin exoskeleton, right? So they obviously they belong to the group of arthropods, the phylum arthropods, and is formed by the polymerization of A. N-acetyl glucosamine, B. lipoglycans, C. keratin sulfate and chondroitin sulfate, and D. D-glucosamine. So it's N-acetyl glucosamine. Okay, chitin is basically polymerization of N-acetyl glucosamine. Okay, yep. So this is a structure of chitin. You see, the N means the number of structures of chitin that are formed. Okay, it's a glycosidic bond that is formed. Chitin is made up of N-acetyl D-glucosamine units held together by beta 1 to 4 glycosidic bond. Okay, so this is a glycosidic bond that is formed 1, 4. Okay, between 1 and 4. This is N-acetyl glucosamine. This is also N-acetyl glucosamine, and a glycosidic bond is formed between them. And N is like the polymerization, the number of different, it could be anything, it could be 5, it could be 10, it could be 100, it could be 200, it could be anything. Okay, this N could be 200 or 1000. Okay, so they are polymers of that. So this is just a way of representing the structure. Okay, so in this case we're talking about chitin. And this is N-acetyl glucosamine. This is also N-acetyl glucosamine, and they are covalently bond together by the glycosidic bond. So it's a structural polysaccharide found in the exoskeleton of some invertebrates. Okay, like in the case of cockroaches. Okay, one good example is cockroach. Okay, their exoskeleton is made of chitin, which is actually N-acetyl glucosamine. Okay. All right, let's move on then. Question number 7. Which one of the following is a non-reducing carbohydrate? Okay, we're talking about non-reducing carbohydrates. Appeared in 2014. A. maltose, B. sucrose, C. lactose, D. ribose 5-phosphate. Okay, so out of the following carbohydrates, B. sucrose is a non-reducing sugar. Answer is B. Okay, so sucrose is a disaccharide as a combination of glucose and fructose. Right? Let's remember that glucose plus fructose forms sucrose. Now, glucose and fructose join together, forming a glycosidic bond, leaving no free aldehyde and free ketone group to react to. All right, so glucose has a free aldehyde group, okay, in its structure, and fructose has a free ketone group. Now, when it's a non-reducing sugar, obviously now we know the sucrose is a disaccharide, so it's a combination of glucose plus fructose. So they're joined together, forming a glycosidic bond. Okay, this is glucose, sorry, and this is fructose. They form a glycosidic bond over here. Okay, they did, so that means that they don't leave any free aldehyde group over here or free ketone group to react to anything else. All right, so when there are no free aldehydes or no free ketone group, that means they are non-reducing sugars. Okay, that means no other reaction can take place between them. Okay, so obviously the combination of glucose and fructose together form sucrose. Okay, so sucrose as such, if you look at the whole structure, is a non-reducing sugar. Okay, when you combine glucose and fructose, forming sugar, forming sucrose. But glucose as such is a reducing sugar, or fructose as such is alone is a reducing sugar. Okay, but in this case we were talking about sucrose. Okay, so sucrose is a non-reducing sugar, hence it is a non-reducing sugar. If we go back to, yeah, which of the following is a non-reducing sugar? Yeah, clearly over here it is sucrose. All right, let's move on then. Question number 8. Macromolecule chitin is, it's appeared in NEET 2013. I think we already discussed chitin before. It's A. sulfur containing polysaccharide, B. simple polysaccharide, C. nitrogen containing polysaccharide, D. phosphorus containing polysaccharide. So it's chitin we know as N-acetyl D-glucosamine. So it is actually a nitrogen containing polysaccharide. Answer is C. Again, we discussed this chitin structure. So yeah, chitin is made up of N-acetyl D-glucosamine units held together by beta 1 and 4 glycosidic bond. It is N-containing, nitrogen-containing polysaccharide. You can see over here. All right, and the structural polysaccharide, we already discussed that before. I guess we can move on then. This was also a repetitive question, right? It's a repeat of the previous question almost. If you look at the discussion, you already know the answer now. Okay, question number 9. Carbohydrates are commonly found as starch in plant storage organs. Yes. Which of the following 5 properties of starch, 1 to 5, make it useful as a storage material? This appeared in 2008. 1. Easily translocated. 2. Chemically non-reactive. 3. Easily digested by animals. 4. Osmotically inactive. 5. Synthesized during photosynthesis. Okay. So now we are looking for a useful property that will be helpful as a storage material, right? See, if something is easily translocated, it's not useful as a storage material. Chemically non-reactive? Yes. Easily digested by animals? No. Not starch as such is not easily digested by animals. Uh, for osmotically inactive? Yes. Synthesized during photosynthesis, but that's not really a property that you can use as storage material. So it's 2 and 4. The answer here is D. Answer is D. Let's do the discussion. So starch is a storage polysaccharide found in most plants. It is made up of two polysaccharides. It's made up of water-soluble amylose and water-insoluble amylopectin. So most of it is insoluble. See, amylose part is water-soluble. It's only 15 to 20% of starch. Okay, and amylopectin is 80 to 85%. Okay, so the insoluble part is more. So that is a good property to store something. Okay, so it is osmotically inactive. Okay, it will remain stable. It will not be chemically reactive with anything also. So they are stored in granules in plant cells. Okay, they're chemically non-reactive and are osmotically inactive. All right, so they are not easily soluble in anything. They don't osmotically participate with water to change the property or the polysaccharide content in it, okay, because of the presence of water. Okay, so they're very stable as such. So these are like good properties to have. Even when we discuss glycogen, I think in previously in one of my lectures, also have similar properties. So glycogen is a animal starch. It's also known as animal starch. So that is a good storage material in animals, whereas starch is or it's also known as plant starch is also known is good for these properties are good for storing energy or polysaccharides in plants. Like glycogen is good for animals. The properties such as they're chemically non-reactive or they're osmotically inactive are good for glycogen. Similarly, it works for starch in plants. Okay, okay, let's go back to the question. This was a good question. So, 1 second. Okay, this research smiley face. This is a good question. Important to understand. Okay, this wasn't just a direct question where you just have your question and you have your options and you can easily choose from it. But yeah, this you had to apply your knowledge into this. So yes, for something that you want as a good storage material, it should be chemically non-reactive. It should be osmotically inactive. These are very important properties that you should have for a good storage material for energy. Okay, so yes, starch in plants, they have these properties and so they behave as good storage polysaccharides. Okay, let's move on. Question number 10. Cellulose is the major component of cell walls of, and this appeared in 2008. A. Pseudomonas, B. Saccharomyces, C. Pythium, D. Xanthomonas. Okay, so cellulose is a major component of cell walls of C. Pythium. Okay, answer is C. Let's go to the discussion. So Pythium is a protist. Okay, protista. Previously it was classified as fungi, but not anymore. Now they are this was long back, but now they are known as protists. Okay, which contains cellulose in its cell wall. Learn this as a I forgot to put this in italics, but yeah, should be underlined here. Pythium, sorry, is a protist which contains cellulose in its cell wall. Learn this as a fact. Saccharomyces contains polysaccharides such as glucose, N-acetylglucosamine, and mannose in its cell wall. Pseudomonas contains peptidoglycan in its cell wall. Xanthomonas contains uronic acid, glucose, and mannose. So clearly the question was which one has cellulose in its cell wall? It's Pythium. All right, Pythium is a protist which contains cellulose in its cell wall. Learn this as a fact. All right, let's move on then. Question number 11. Carbohydrates, the most abundant biomolecule on earth, are produced by, this appeared in 2005, A. Some bacteria, algae, and green plant cells. B. Fungi, algae, and green plant cells. C. All bacteria, fungi, and algae. D. Viruses, fungi, and algae, and bacteria. I'm sorry. So viruses and fungi don't produce bacteria. So B, C, and D are wrong. It's A. The answer is A clearly. Answer is A. There's some bacteria, algae, and green plant cells which produce carbohydrates. Okay, please remember again this as a fact. You need to know what produces carbohydrates. Yes, some bacteria produce carbohydrates. There's algae which produces carbohydrates, and of course green plant cells. All right, let's move on then. Question number 12. Which of the following is a reducing sugar? This appeared in 2002. We spoke about non-reducing sugar. Now, which one of them is a reducing sugar? You have A. galactose, B. gluconic acid, C. B. methyl galactoside, and D. sucrose. Clearly, it's not sucrose. It's not beta methyl galactoside or gluconic acid. It is galactose. Okay, so the answer is A. So galactose is a galactose is a monosaccharide and is found in lactose, and galactose is a reducing sugar since it has a free aldehyde group. Okay, so you need to remember this as examples. Like I said, sucrose is a non-reducing sugar and galactose is a reducing sugar. Okay, since it has a free aldehyde group. Okay, so this is a way of identifying which sugars are reducing sugars and which are non-reducing sugars. All right. Okay, I guess we can move on. Okay, so question number 13. Cellulose, the most important constituent of plant cell wall, is made up of, and this appeared in 1998. A. branch chain of glucose molecules linked by beta-1,4-glycosidic bond in straight chain and alpha-1,6-glycosidic bond in the site of branching. B. unbranched chain of glucose molecules linked by beta-1,4-glycosidic bond. C. branch chain of glucose molecules linked by alpha-1,6-glycosidic bond at the site of branching. D. unbranched chain of glucose molecules linked by alpha-1,4-glycosidic bond. So, cellulose is unbranched. Okay, so A and C are out. Also, D. unbranched chain of glucose molecules linked by alpha-1,4-glycosidic bond. That is also incorrect. So, it is B. Okay, the answer is B. Let's go to the discussion. So cellulose is an unbranched chain of glucose molecules linked by beta-1,4-glycosidic bond. Okay, so this is a structure of cellulose. Okay, so you have the beta glucose, you have glycosidic bonds formed here. Yeah, and yeah, this is 1 and this is 4, so there's 1,4 linkage, 1, 4 and so on. All right, so this is the structure of cellulose. I would suggest you remember it, okay, because they've asked a question on what the structure of cellulose is, so you need to be aware. Okay, all right, so let's move on. Question number 14. Lactose is composed of, and this appeared in 1998, A. glucose plus galactose, B. fructose plus galactose, C. glucose plus fructose, D. glucose plus glucose. Okay, so lactose is composed of glucose plus galactose. Okay, so the answer is A. Okay, so here you have a structure of lactose, which is also known as milk sugar. So if they even write milk sugar, you know they're talking about lactose. Okay, so combination of beta-D glucose and beta-D galactose linked by beta-1,4-glycosidic bond. So this is a structure of galactose, structure of glucose. And this is the one, this is the 4th position over here. And they're linked by the glycosidic bond. Okay, but you need to know, yes, a disaccharide, and it's a combination of galactose and glucose. All right, so that's what they were pretty much asking in the question here. Okay, we can move on. Question number 15. In which of the following groups are all polysaccharides? Okay, and this appeared in 1996. A. Sucrose, glucose, and fructose. No. Maltose, lactose, and fructose. No. Glycogen, sucrose, and maltose. No. Glycogen, cellulose, and starch. Yes, this is a good option. So the answer here is D. Okay, glycogen is also polysaccharide, cellulose is a polysaccharide, and starch is also a polysaccharide. All right. So polysaccharides are monomers of monosaccharides. So this is the definition of polysaccharides. Polysaccharides, they are monomers of monosaccharides with a high molecular weight. They are tasteless and form colloids with water. Okay, there are two types: homopolysaccharides and heteropolysaccharides. I've given some examples here of homopolysaccharides and heteropolysaccharides. So if you want, you can note them down. But just remember that the definition of polysaccharide is this. They are monomers of monosaccharides. It's very important to remember, okay, with a high molecular weight. If we go back to the question, the options given below, it was not sucrose and maltose, although they are disaccharides. But clearly option C was wrong. The correct option here was glycogen, cellulose, and starch. Okay, all of them are polysaccharides. They're monomers of monosaccharides. Okay. All right, I guess we can move on. Okay, here we have a structure of amylose and amylopectin. Okay, it's a starch. Okay, let's move on. I think we discussed starch also before, right? So yeah, this is the structure of starch here, amylose and amylopectin. So clearly we can see it's made up of glucose. All right, let's move on then. Question number 16. Glycogen is a polymer of, this appeared in 1993. A. galactose, B. glucose, C. fructose, D. sucrose. So we just discussed polysaccharides. So glycogen is also known as animal starch. Okay, and it's a polymer of B. glucose. Answer is B. So glycogen, animal starch, is a polymer of glucose. Glucose are held together by alpha 1,4 glycosidic bond and alpha 1,6 glycosidic bond at branching points. Okay, so this is 1,4 glycosidic bond here, yeah, and this is 1,6 at the branching chain. Okay, over here, yeah, this portion over here. Okay, so yep, this is the structure of glycogen. Okay, I would suggest you remember it, that there are branched chains over here in the case of glycogen. So which is branched and which is unbranched? Like cellulose is unbranched. Do you remember that? Starch is branched. Glycogen is branched. So I would suggest you remember these structures, which is branched, which is unbranched, which is alpha, which is beta. You know, so yeah, they could possibly ask you that in the exam. All right, but if you look at the question, it was pretty simple. Glycogen is a polymer of B glucose. Okay, let's move on. Question number 70. Which of the following are not polymeric? This appeared in NEET 2017, pretty recent. A. proteins, B. polysaccharides, C. lipids, and D. nucleic acids. So, are not polymeric. Okay, so the answer here is C. lipids. The rest are polymeric, A, B, and D. C. lipids is not polymeric. Okay, remember that answer C. So lipids are non-polymeric in nature. Okay, now polymer, you know, we've seen carbohydrates, we've seen proteins. They're polymers of these amino acids. and nucleic acids the nucleic acids are polymers of these nucleotides. But in the case of lipids, they're not polymeric in nature, unlike proteins, nucleic acids, and carbohydrates. Instead, lipids are made up of fatty acids attached to an R group. Okay, this is how they are structured in nature, okay, naturally. So they don't form polymers of anything. Okay, so they're just made up of fatty acids and they're attached to an R group, a variable group. That's all. Okay, this was a good question. This was important to remember which of them are polymers and which are not polymers. So clearly lipid is non-polymeric in nature. Good question. Let's move on. Question number 18. Nucleotides are building blocks of nucleic acids. Each nucleotide is a composite molecule formed by, this appeared in 2005. So we're talking about nucleotide. Okay, it has A, base sugar phosphate, B, base sugar OH, C, base sugar phosphate with the N, and D, sugar phosphate. Okay, so nucleotide is a composite molecule formed by I'm sorry, it's not C, it's A, base sugar phosphate. Now what is the difference between A and C? Okay, now there's an N group, that means a N, like a number of molecules that it can have, but C actually forms nucleic acids. Okay, and A, base sugar phosphate 1 forms nucleotide. Okay, this is a nucleotide. This is a very important difference that you should know, and this forms nucleic acids. So it's a polymer of nucleotides, right? That's C, but A, base sugar phosphate as such forms nucleotide molecule. Okay, so the answer here is A. Okay, so here you have a structure of a nucleotide. So nucleotides are composed of base, pentose sugar, in this case it's ribose sugar, and phosphate group. Okay, here it's a phosphate group. Okay, so this is a typical structure of nucleotide. Okay, let's move on then. Okay, so question number 19. Which purine base is found in RNA? This appeared in 1996. A, thymine, B, uracil, C, cytosine, and D, guanine. Now they're talking about which purine base, not pyrimidine. So uracil is pyrimidine, so B is out. Cytosine is also pyrimidine, C is out. Thymine does not appear in RNA, and again it is a pyrimidine. Adenine and guanine are purine. Okay, this is a good twist. It is very easy to look at RNA and then identify B as, the one that appears in RNA. No, they're talking about which purine base. Okay, so clearly guanine, yes, appear in RNA and DNA as well. But which purine, if you just look at the question alone, which purine base is found in RNA? Of course, guanine. Adenine is not there in the options. So A, B, and C are incorrect. The answer here is D. Again, it's a very good twist to the question. If you don't read the question properly and you are not able to differentiate between purine and pyrimidine bases, you get the answer wrong. Okay, let's go to the discussion. So guanine is a purine base that is found in RNA as well as D and DNA. Okay, so this is the structure of guanine. Okay, let's move on. Question number 20. Which of the following nucleotide sequences contains 4 pyrimidine bases? This appeared in 1994. Okay, talking about 4 pyrimidine bases. Okay, we're talking about pyrimidine as such. So it can't be A or G. It has to be either U, C, or T. Okay. We don't know whether it's a DNA sequence or an RNA sequence. It's not mentioned here. Okay, so we just know that's U, C, or T. And we have T here, we have T here, we have C here. So there's only 3. We have a C here, we have U here, we have C here. Again, only 3. We have U here, we have C here, we have, oops, sorry, U here. Okay, I think I'm missing out somewhere. Oh yes, there's a C here over here. So C, option C and option B are wrong. If you look at option A, you have T, you have C, you have T here, and again you have C here. So it's a DNA sequence. And that's how you know that this is a DNA sequence, but there's no uracil in it. Okay, so the correct answer here is A. Okay, it can't be B, it can't be C. Obviously, the option is not D. It has to be A. So it has 4 pyrimidine bases in it. Okay, this is how you identify the bases in the sequence. Okay, so pyrimidine bases are thymine, cytosine, and uracil. Okay, so option A contains 4 pyrimidine bases. We just saw that it was a DNA sequence, and obviously uracil is absent. In place of that, you have thymine. Okay, thymine and cytosine. All right, let's move on. Question number 21. In RNA, thymine is replaced by, this appeared in 1992, A adenine, B guanine, C cytosine, and D uracil. We all discussed this before. So in RNA, thymine is replaced by D uracil. Okay, the answer is D. This was pretty simple. So in RNA, the bases are A, U, G, C. Okay, in DNA there will be T. In RNA it will be uracil. This is for DNA. Okay. Yeah, so in RNA, thymine is replaced by uracil. Alright, this was pretty direct. Okay, so we've come to the end of this video lecture. And I hope you've enjoyed today's video lecture and hope you've learned something new today. So don't forget to like, share and subscribe to NEET Biology. And I'm available on Facebook, Twitter, Instagram. So you can follow me there for more updates. And I'll see you in the next video lecture, right? So bye.